So, hello guys. Today we're going to discuss a respiration plan. So, this will be a one short, video on or one short class on respiration plans. We will be discussing all the important topics from this chapter uh, in a one short video, and also we will be working out, important MCQs based on that. Okay. So let's start. I hope my slide is visible for you guys. Okay. So I'm starting one by one. So in respiration, we know that respiration is an important, chapter. Um, Here we will be discussing different topics like, glycolysis, Krebs cycle, electron transport system, um, you know, respiratory quotient. All these topics we will be discussing on this chapter. So let's start one by one. Okay. So let me take the first. So the chapter name is Respiration in Plants. So during this chapter, we will be discussing mainly about, glycolysis. Then we will be discussing about, Krebs cycle. Then we will be discussing about electron transport chain, and also we'll be discussing about oxidative phosphorylation. So these are the different steps in respirations. Okay. So let's take one by one. When you take respiration in plants, first we will be discussing about glycolysis. Okay, fine. So when you take glycolysis, you know that what is the meaning of glycolysis? Glycolysis means during glycolysis, one molecule of glucose will be converting into two molecules of pyruvic acid, right? So this is the glycolysis, the first step during which glycolysis is the first step where respiration will be starting. Okay, give me a minute. Meanwhile, let me set a few things. Give me a minute. Okay, so yep, and whoever joined, just say hi in the box. Okay. Yep, 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 yep. So here first we will be discussing about glycolysis, then we will be discussing about Krebs cycle, and also we'll be discussing, important questions related to this. Okay, fine, 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 fine. So yep, yes, fine. So here you can see that in glycolysis, what exactly happens. So glycolysis means, the meaning of glycolysis is that here during glycolysis, glucose will be converted into pyruvic acid. Pyruvic acid. How many molecules of pyruvic acid? There will be two molecules of pyruvic acid, right? And this is called the glycolysis. So after glycolysis, either pyruvic acid may enter into aerobic respiration or anaerobic respiration. If it is anaerobic respiration, then it will be like, say, pyruvic acid will be converted into, let's say, C2H5OH, which mean alcohol, right? In pyruvic acid, there will be 3 carbon. In ethanol or C2H5OH, there will be only 2 carbon, which means there will be one excess carbon, which will be released in the form of carbon dioxide, right? Now, here there are 2 molecules of pyruvic acid. So, there will be formation of 2 molecules of alcohol and 2 molecules of carbon dioxide, right? So, and this process is called alcoholic fermentation. Alcoholic fermentation, right? And another possibility is that this pyruvic acid could be converted into lactic acid. How many carbons will be there? 3 carbons will be there, right? Because pyruvic acid 3 carbon, lactic acid also 3 carbon, which means both are having equal number of carbon, which means there won't be any excess carbon, so there won't be any release of carbon dioxide, right? So, and this process is called lactic acid fermentation. Lactic acid fermentation. Now, so what happened right now? Glycolysis has happened. Glycolysis means glucose is converting into pyruvic acid. This glycolysis is a common process in both aerobic and anaerobic respiration. In both aerobic and anaerobic respiration, the first step will be, or first process will be glycolysis, right? So after glycolysis, if it is an anaerobic respiration, after glycolysis, they will undergo anaerobic respiration or fermentation. See, this is one possibility. When you take yeast, in yeast will show alcoholic fermentation, where the product will be alcohol as well as carbon dioxide. So when you take muscles, in our muscles, there will be lactic acid fermentations, right? So let's say we are running for long. So you can see that after a time, we will start getting some pain in our muscles, which is because of the release of lactic acid, right? So we will see one question based on this concept. Let me take one question. Yeah. So here one question from 2014 NEET exam. In which one of the following process carbon dioxide is not released? Lactate fermentation, aerobic respiration in plants, aerobic respiration in animals, alcoholic fermentation. So what will be the answer here? We know that carbon dioxide is not released in lactic acid fermentation, right? So option A will be the answer. Yeah. So it's very clear from here. You see, in lactic acid fermentation, there is no release of carbon dioxide, right? Now, let me get into glycolysis, right? I hope you guys are here. Just say hi in chat box so I can understand that. Okay. So, glycolysis. Okay. So, when you take glycolysis, you can see that first of all, we will start with the glucose. In glycolysis, glucose will be converted into, first of all, glucose will be converted into glucose 6-phosphate. Glucose 6-phosphate, right? So, how many carbons will be there? Glucose will have 6 carbons. Glucose 6-phosphate will have again 6 carbon. But here you can see that, here you can see an extra phosphate at a 6th position. This extra phosphate was given to glucose by carbon dioxide, which so it means that during the first step, one extra carbon dioxide is added. One extra carbon dioxide is added, right? That's why glucose is converting into glucose 6-phosphate. Later, this glucose 6-phosphate will be converted into fructose 6-phosphate. And you can see these two are isomers. And one important thing, during the first step, which means the conversion of glucose to glucose 6-phosphate, this process is catalyzed by an enzyme called a hexokinase enzyme. What is that? Hexokinase enzyme. It was a PYQ in NEET exam. Very, very important. Later, this fructose 6-phosphate will be converted into fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. And later you can see that this fructose 1,6 bisphosphate will be divided into two molecules. One is called dihydroxy acetone phosphate. Dihydroxyacetone phosphate. And the another one is called glyceraldehyde glyceraldehyde 3-phosphate. And these two compounds are interconvertible. These two compounds are interconvertible. When you see the number of carbons, here you can see that glucose has 6 carbon, glucose 6-phosphate has 6 carbon, fructose 6-phosphate has 6 carbon, fructose 1,6 bisphosphate has 6 carbon, but dihydroxyacetone phosphate will have 3 carbon. Hence, it is a triose sugar. Glyceraldehyde 3-phosphate will have again 3 carbon. So these two are triose sugar. Later, what happens is that this glyceraldehyde 3-phosphate, this glyceraldehyde 3-phosphate, here you can see that later this glyceraldehyde 3-phosphate will be converted into another one. Let me write it here. So I will write this step separately here for better clarity. Here you can see that fructose 1,6 bisphosphate were there. I'm repeating the same step. Okay. And then it is dividing into two compounds, right? These two compounds are called what? One is called dihydroxy acetone phosphate. Another one is glyceraldehyde 3-phosphate, right? And these two are interconvertible, right? To avoid the confusion, let me erase this. Okay, otherwise it may create confusion later. Okay, so this compound converted into fructose 1,6 phosphate, then dihydroxyacetone phosphate. And during this conversion, what happens, guys? During this conversion, you can see that when fructose 6-phosphate converted into fructose 1,6 bisphosphate, you know that fructose 1,6 bisphosphate has one extra phosphate at the first and sixth position. Here there will be two phosphate. Here they have fructose 1,6 phosphate has only single phosphate. So what happened during this conversion? During this conversion, um, extra phosphate will be added by ATP. I think I made a mistake. During first step, which means from glucose to glucose 6-phosphate, what will be adding, guys? There will be addition of ATP. Because here the number of phosphates are changing now. Glucose has one phosphate, but sorry, glucose has 0 phosphate. Glucose 6-phosphate has one phosphate. So during this conversion, extra phosphate will be added. Extra phosphate will be added. Okay, fine. Now, later what happens, guys? uh, this compound is dividing into two, that is dihydroxyacetone phosphate and a glyceraldehyde 3-phosphate. Later, this glyceraldehyde 3-phosphate will be converted into 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate. Right? So during this time, what happened, guys? Extra phosphate added because glyceraldehyde 3-phosphate have 1 phosphate. 1,3 bisphosphoglycerate has 2 phosphate. Here 1 phosphate, here 2 phosphate. So during this conversion, extra phosphate added, and this extra phosphate will be given by inorganic phosphate. By this time, NAD plus will be converted into NADH plus H plus, NADH plus H plus, right? Later, this 1,3 bisphosphoglycerate will be converted into 3-phosphoglycerate, 3-phosphoglycerate, and 3-phosphoglycerate will be converted into 2-phosphoglycerate, which will further converted into phosphoenol pyruvate, and finally it will form pyruvic acid. See here, you can see that when 1,3 bisphosphoglycerate converting into 3-phosphoglycerate, you see here this compound is having, see this compound is having 2 phosphate, but 3-phosphoglycerate has only 1 phosphate. Correct now? 1 phosphate. So during this conversion, what happened exactly? 1 phosphate has released. This released phosphate will be accepted by ADP, and ADP will be converted into ATP. Correct? So in the first process, what is happening? When you take the initial steps, you can see that here ATP will be converted into ADP because ATP is using. Hence, ATP is converting into ADP. Even in here as well, you can see that what is happening here? ATP is converted into ADP, right? But here at this stage, ADP will be forming ATP, right? Then when 3-phosphoglycerate converting into 2-phosphoglycerate, you can see that there are no change in phosphate number. Here 1 phosphate, even here there will be only 1 phosphate. Later, 2-phosphoglycerate will be converting into phosphoenol pyruvate, again 1 phosphate. Finally, phosphoenol pyruvate converting into pyruvic acid, right? You can see that during this pyruvic acid has 0 phosphate. So during this conversion, 1 phosphate has released. That released phosphate will be accepted by ADP, and ADP will be converted into ATP, right? Fine. Now, when you see the number of carbons, you can see that now we're going to check the number of carbon. Glucose 6 carbon, and this compound has 6 carbon. This compound has 6 carbon. This compound has again 6 carbon. Now this is 3 carbon compound, and this one is 3 carbon compound. This one is 3 carbon compound. This one is 3 carbon compound. Again here 3 carbon. Here 3 carbons. Here 3 carbons. Right? Now you see. Now here you can see that how many pyruvic acids are formed here? Only 1 molecule of pyruvic acid has formed here. But as per the definition, we know that glycolysis means it will form 2 molecules of pyruvic acid. So what happened here, in here, the first molecule of pyruvic acid, the first molecule of pyruvic acid has formed from glyceraldehyde 3-phosphate. Later, what will happen? Dihydroxyacetone will be converted into glyceraldehyde 3-phosphate. After the second cycle, which will form second molecule of pyruvic acid. Second molecule of pyruvic acid. Right? So there will be formation of two molecules of phosphoenol pyruvate, two molecules of 2-phosphoglycerate, two molecules of 3-phosphoglycerate, two molecules of 1,3-bis phosphoglycerate. So this is how it works. So when you take glycolysis, here onward, there will be two cycles. But above, when you take the upper part will have only one cycle. I hope it's clear. Now let me calculate the ATP, NADP, ATP and all. Okay. So when you take glycolysis, see here how many ATP has formed here? ATP formed. Here you see, here, yeah, see here one ATP has formed. Here, oh my God, I made a mistake here. Here nothing but here, yeah, correct only, na? Correct only. ADP to ATP. So here, second molecule of ATP has formed. But here one ATP, here one ATP. But it is in one cycle now. If there are two cycles means what? 2 into 2. 2 into 2 ATP. Means total there will be 4 molecule of ATP. 4 molecules of ATP will be formed. And out of this, how many ATPs are used? That is a real question. See, here one ATP used, here second molecule of ATP used, right? So out of 4, you can see that 2 ATP used. So what is the net gain? Remaining will be 2 ATP. 2 ATP will be the net gain, right? Now, how many NADH2 has formed? How many NADH plus H plus has formed here? See guys here. When you take NADH2, let's say here one NADH2 has formed, which is in one cycle. If there are two cycles, then there will be two molecules of NADH2, right? So we can say that two molecules of NADH2 in one glycolysis, two molecules of NADH plus H plus will be formed, right? And we know that one NADH plus H plus or one NADH2 is equal to 3 ATP. So 2 molecules of NADH2 means what? 2 into 3, which is 6 molecules of ATP. So if you combine everything in terms of NADH2 as well as direct ATP, then totally how many ATP will be here? 6, 6 ATP from here, then 2 ATP here. So 6 plus 2, there will be 8 molecules of ATP. And if you combine whole ATP, let's say initial 4 ATP, then 6 ATP, 6 plus 4, 10 ATP. Out of this 10, 2 ATP has used. So what is remaining? 2. So 6 plus 2, 8 ATP. Okay, this is the mathematics works. So we will be, for this class, we will be discussing only glycolysis. I will work out questions also based on this topic. One shot we will do some other day since many students are, couldn't join today due to some technical issues. Okay. Yeah. So this video will be just discussing only glycolysis and fermentation, and we will do questions as well. And we will plan live, I mean, one shot on this chapter in some other days. Okay, fine. So let me take, yeah, question. Assertion, ATP is used at two steps in glycolysis. A reason, first ATP is used in converting glucose into glucose 6-phosphate, and second ATP is used in conversion of fructose 6-phosphate into fructose 1,6 diphosphate. So when you take this assertion, ATP is used in two steps in glycolysis. This is true because you see, ATP has used in two steps. Say they used here as well as here. So at two steps, ATP has used. True only. Now, what was the reason? First one is glucose into glucose 6-phosphate. See, first stage is what? Glucose into glucose 6-phosphate. Here ATP has used. What is second one? Fructose 6-phosphate into fructose 1,6-phosphate. So at the two stages, ATP has used, right? So this also true. So both are true, and as reason is the correct explanation for assertion. So option B will be the answer, right? Next one. What is the net gain of ATP when each molecule of glucose is converted into two molecules of pyruvic acid? Means they are asking uh, during one glycolysis. During one glycolysis, what is the net gain of ATP? We know that it will be 2 ATP. We have already discussed before, right? So option D will be the answer. Next, conversion of glucose into glucose 6-phosphate. The first irreversible reaction of glycolysis is catalyzed by aldolase, hexokinase, entolase, phosphofructokinase. Very easy. We already discussed. It will be hexokinase enzyme, right? That's it. So by saying this, I am ending this live session. So make sure, I hope you understood it properly. If you have any doubt, you can, you can let me know your doubts in comment section, and also you can join our Telegram group. You can get the lecture, slides and everything. And also let me know how the class was, how much you understood. I would like to know your feedback as well. So see you soon. Okay, bye. Take care.